Welcome to the Explain podcast, a podcast where next generation sequencing techniques are explained to me by Daniel.
That's me, because I'm a bioinformatician and Iuliia is a physicist who joined us recently at the sequencing facility. We are two colleagues from the Next Generation Sequencing Competence Network and we work at the West German Genome Center in Düsseldorf.
Hello everyone! And now the podcast Explain once more with you. After receiving feedback, we realized that we need a fundamental episode.
Where we really explain the fundamental words that we will say here every 20 seconds, that everyone can go along with what we explain and understands where we are going with it. So we start with the most fundamental dogma of biology, which is we go from DNA to RNA to protein.
Oh yeah, that sounds absolutely fantastic. And you know what? I will start explaining it to you. So if you think it's stupid, give us a feedback, but be gentle about it.
And since I'm a bioinformatician and Iuliia is a physicist, I think it will be the most fun that Iuliia starts with explaining stuff.
Why not? Really?
Okay, Iuliia.
For sure, I Googled. You know about it, right? To not be super basic stupid. What we all know about DNA. Daniel, do you know what DNA is? Like this deoxo...
Mm-hmm.
Tell me about it. Deoxyribonucleic acid?
Yeah.
Something like that.
Yeah.
So I definitely know there is this ribonucleic acid, which is RNA.
Mm-hmm. And I will talk about RNA.
But I already start telling you that it's a single-stranded material and DNA then a double-stranded material.
We will talk about that. It's not that easy. There are certain kinds of RNA, there are several kinds of DNA. So let's just start with
now it's really
DNA. Yeah.
What is
interesting? What is that?
So I'm now starting to draw the double-stranded DNA, genetic material, what and why it is in our bodies, in all creations of life. It carries information. But which kind of information, Daniel? I know it also from a textbooks. DNA carries an information. And I was like, okay, wonderful.
Yeah. So you already started with the double helix, yes?
Yes.
That is very central to the DNA, to double-stranded DNA, I should say. But the information itself is not about the strands, it's about the bases between the strands.
So the bases, people, bases are these tiny structures in between the double strands. Are both strands RNA or one is RNA and another one something else?
No. So the lines that you made here, we call it the DNA backbone. The line that goes from A to B here, that is a chain of sugars.
Oh, hello there. Sugars.
That's the sugar backbone. That's the deoxyribose. Right?
Oh, wonderful. Okay, yeah, I've heard that one.
That is the backbone that carries the information. And the information is the bases that are between two of those strands, or one. There is even a type of single-stranded DNA, but we will come to that later.
Oh, yeah, please.
The most native, the most abundant form of DNA is that double-stranded helix here that you can see in a lot of logos and stuff. There are 4 different bases that can be on top of the sugar.
I know. These are nucleotides, right?
Exactly. Good point, Iuliia. Can you tell me which nucleotides? Because there are multiple. There are more than 4, but DNA has only 4.
They are more than 4, but oh God. These are these CTA... Nanana...
Nanana. If nanana is a G, then yes.
Yeah. Can you name them for us? Because for me, it's a bit complicated, of course, to get to it.
Adenine.
Yeah, adenine.
Guanine.
Cytosine.
Yeah, and thymine.
Thymine.
Yes.
So people, here we go.
Okay, and there is a central property of DNA, which is that only 2 of those bind to each other every time. So a C cannot pair to T or A or G.
Like never.
Like never.
Is there a big reason why?
So there are 2 bases. 2 of those bases are connected by 2 hydrogen bonds, and the other 2 bases are connected by 3 hydrogen. And only the 3 hydrogen bonds can bond to each other, and the 2 hydrogen bonds acids. So which one goes to which?
Yeah, exactly. You said that this one, the
cytosine.
Cytosine? Okay, you said that the C part is cannot be connected to anyone.
Exactly. So the C cannot connect to T or A or G. It can only connect to one.
Only to one from these ones.
Only
Not
to one.
not everything at once. That's what you meant. Okay. DNA base, it's like a building block.
It's like a letter in an alphabet, or if you compare it to how you store data on a computer, it's like ones or zeros, but just you have 1, 2, 3, 4, basically.
And with these 2 or 3 hydrogen bonds, does every base can have 2 or 3 hydrogen bonds, or each of them have some specific properties? For example, the C base can have only 2.
So T, thymine, and A, they can only have 2. They have 2 hydrogen bonds, which means only they connect to each other. So T, A. And if you have on the other strand an A, then you can expect the other strand to have their T. Okay?
Okay.
Okay. And the other case is with C. What do you think?
Well, now the G is only what's left.
Exactly. The next thing I wanted to explain is that the sugar, the backbone here, it's a desoxyribose. That is where the name comes from. Okay? So it's a sugar with, phosphorus.
Yeah.
And you have typically, there's the base, let's say an A, which has a different structure. We will come to that later.
The A, this nucleotide A.
For example, yeah, exactly. One of those bases. And on this connection here, there is typically an OH group, if that would be the end of a DNA strand. If not, then this is the connection to another P.
To another P, okay.
And this P is then connected to another sugar. Right? And there comes another P.
And there comes another sugar.
Sugar, phosphorus, sugar. That is what this backbone here is on both strands. It's the same.
Okay, this is kind of this single strand. So the sugar has normally 5 endings.
Desoxyribose, yes.
Exactly, this sugar. So desoxyribose has 5 endings. And we discussed
5C, 5 carbon.
Yeah, 5
edges,
carbon
let's say.
Connections. Connection points. And we said that one has connection to a nucleotide, another one phosphorus, and another one phosphorus. And phosphorus are the connection points to the next sugars.
Yeah.
What's happening with another 2 free connection carbon points that you left out free?
You are a chemist. Okay. So what happens if you have 5 corners?
They are vacant, aren't they?
And you have, let's say, C's here. What happens in nature if you would have just this molecule?
Yeah, they would try to connect to something else.
Or themselves multiple times. The phosphorus is a really important part for the DNA because it links one part, basically one connection to a base, to the other, to the next. And if there is, at some point, there is an end of DNA. And then there is an OH group on the one end. And on the other end, there's then an open phosphorus group.
All right.
Okay. That is important for all sequencing techniques we are talking about in the future. Okay.
Because you're using this OH group, something like a label or a marker for the ending, right?
Or beginnings or ends of enzymes that dock on and start doing something.
Okay. Hello, enzyme. Explain.
We will start with the most basic part. Enzyme is a chain of amino acids. It mostly is just a bulky little thing. Like most enzymes have somewhere in the sequence. So it's a really long strand of amino acids, different amino acids. There are more than 4 amino acids. Okay. And at some point, there is in an enzyme typically an active site, let's say a magnesium, which is bound to the amino acid structure. And that is then the active site of this enzyme. And this catalyzes a reaction. What is catalyzation? Do you know a catalytic converter on your car?
Catalysis should improve somehow the speed of reaction in a way, or make it more effective?
Yeah, okay. That's sometimes in biology, you have a reaction that goes with a really slow and steady state. So here you have time, and here you have % of conversion.
All right.
Or % of reaction happening. Yeah? And it slowly goes up. But with an enzyme, it does the same reaction, but it makes it faster, okay? So sometimes those enzymes need energy. They sometimes need cofactors. They sometimes need other things like water to work in. But they generally, if you add enzymes at some point, the reaction goes much faster, okay?
Yeah.
With most reactions, you can also say that if you add less enzymes, it goes less faster. And if you add more enzymes, it goes more faster.
Can there be a point when you put so many enzymes that they basically quench the whole reaction?
Yes, that's possible. You can also have that you saturate at some point, that you don't have enough energy for all enzymes to work. But there is also some point where the concentration of enzyme is really high and the amount of things that you want to react is so low that no enzymes finds any more substrate to react with. But typically what we will talk about a lot is that we have some enzyme that's genetically engineered. We will talk about genetically engineered enzymes.
All right, yeah.
And it started out with an enzyme that someone found in a bacteria that does a certain job specifically well, or in specific circumstances, like, for example, a DNA polymerase that does its reaction in really high temperature water, where other enzymes would denature already. But this enzyme is somehow surviving high temperatures, which makes it really handy if you want to control a reaction by temperature, okay?
Yes. All right. And enzymes, their basic function to be catalyzers or not?
Like the catalytic converter on your car. Yeah,
They are.
they are.
Are they also cutting the DNA strands into pieces?
Some, yeah.
Some of them, but not all of them. So there are different types of enzymes as well?
There's 1,000 different types of enzymes which are catalytic converters to a lot of reactions. Okay, so all your metabolism is based on enzymes. Without enzymes, your body wouldn't work. There are some exceptions, as always in biology. For example, there are specific RNAs which behave like enzymes.
RNA behaves like an enzyme.
Sometimes, yes.
How can it be possible? Yeah, it's containing different type of sugar. Enzyme consists of amino acids, right?
Yeah, bound together.
Yeah, and RNA is a single-stranded,
Not always.
this sugar with nucleotides.
Yeah, it doesn't have anything to do with being an acid or not. It just has to do with the structure and whether there is a pocket somewhere where a reaction can be catalyzed.
Could you explain me in a couple words this beautiful pocket? How to say, okay, I have RNA and there is a pocket in there?
Yeah, sure. Okay. As with amino acids, you can also have RNA like this in a single strand, similar to DNA. Okay, DNA, we started like the double strand with the bases in between. And RNA, let's say, is here single-stranded with other bases.
And it has an open basis in a way.
It has an
Looking into an
OH
open
end
space.
and a phosphorus end. I will spoiler this. But also, RNA is more unstable compared to DNA.
Yeah, it also does look much more unstable because once you have double-stranded, they kind of touch nucleotides from both sides. Yeah,
it's the hydrogen bonds. Yeah, and for RNA, you sometimes have long RNAs where the sequence of the nucleotides dictates how the RNA will form into a ball. Let's call it the RNA ball. Okay.
Yeah.
And this RNA ball can also have a pocket, can also have an active site.
It's an empty space within this conformed space, like a ball of RNA. Is there an empty? How can we say it? It's like in the middle of donut there is a space for some sauce, for some jam. Can we have this alignment? So is that a pocket you're talking about within the RNA?
Okay, we will start with something really easy. Okay, we start with transfer RNAs, tRNAs. So the central dogma of biology is that we start with DNA with the bases. Here again, double-stranded, CTHE. Yeah. Then we will all talk about those processes. We go from DNA to RNA, single-stranded at this point.
Yeah.
This has a cap structure and a poly A tail. We will talk about this later. And from this, we go to amino acid chain.
How?
Good question. One step in the amino acid chain synthesis is that there are tRNAs.
All right, yeah.
Okay, so here are 3 ends that can actually pair to the single-stranded RNA. Okay, in RNA, you have C, A, U, and G. What do you remember from DNA?
C can connect with G with the 3 hydrogen bonds and...
Aha!
No, there are... These are other things. Where is T?
Yes, exactly. Good point.
There are no T.
Yeah, but there's G, A, T, C. Okay, so from DNA, we have those 4 bases. We go from DNA to RNA.
What is the U base?
It's RNA specific.
Uracil or something like this?
Exactly, it's an uracil.
I just heard this name, so it was somewhere inside of my mind, but what is it? And like, wow, okay.
So only based on the sequence and whether you find a T or an U, you can determine whether you have RNA or DNA in your hand.
It's not about this. You started to tell about transfer
RNA.
Transfer RNA.
And
isn't it a difference between RNA and transfer RNA with this U and T or not?
Transfer RNA does also have no T, like RNA.
Okay.
That's why it is an RNA and not a transfer DNA.
Oh my God. Okay. All right. No, very interesting. I mean,
Okay.
I'm not sure that it's really clear for many people because we all know these 4 bases, at least not really by their full names, so to say, but there is a C, there is a T, G, and A. Most of the people can tell you that. But then that there is a difference to RNA when you have U instead of T. Huh. All right. Very nice. I've learned something absolutely new today.
Congratulations. Okay, so we were somewhere different. So we started with DNA, GATC. We go to RNA, GAUC. And the transfer RNA are a special kind of RNA where the RNA has like those 3 fingers, let's call them. And those can be complementary to the single-stranded RNA. Right? So if you have a G, this transfer RNA has a C. If there's an A, the complement to an A in this case is an U. And if you have the RNA with a U, the complement is an A. For each of those sequences of the transfer RNA, there is one specific amino acid attached to it. Yeah, so this transfer RNA has an S attached to it. Okay, this is DNA, and this is a tRNA.
tRNA, transfer RNA, and M?
Messenger RNA, because it is the message from the core of the cell to the protein synthesis machinery.
And you tried to sneak this label just like that.
Sorry.
Before you always called it just RNA, and now we have a messenger RNA. So people, there are 2 types of RNAs right now.
No, there are several.
Even more.
There are long non-coding RNAs. What do you think they are?
For the long coding.
Long non-coding, yeah. Those are
non-coding.
Those are long pieces of RNA that don't code. They are not made into a protein at a later stage, but they have other functions. Then you have microRNAs, which are smaller, around roughly 20 nucleotides.
Okay.
And they sometimes bind to mRNAs.
Can we say how many nanometer long they are when you're talking about nucleotides, like 20 nucleotides, how many nanometers it is, or picometers?
We can, but I don't know how long a microRNA is. It's about 20 nucleotides. That is what I can say in length. So
yeah, that's the language of bioinformaticians. That's not the physicists.
We don't speak in meters, we speak in bases.
Yeah, you speak in bases. We have a different currency in our worlds, you know?
Yeah, that's true. Those microRNAs, sometimes they bind to single-stranded mRNA. And when they are bind to mRNA, then the transfer RNA can't dock to it.
Yeah, of course.
And it's then not converted into protein, right? Because if the tRNA cannot dock onto it and bind the amino acid to it, and the amino acid then gets made into a long amino acid chain, and every 3 RNA nucleotides get made into one amino acid. This becomes a long chain, and this long chain at some point becomes a protein. The microRNA thus are inhibiting this function. They are inhibiting protein synthesis.
Why do they even exist? Because RNA is made for...
No, that is exactly what they're made for. MicroRNAs inhibit protein formation.
Why would you want to prohibit protein formation inside of the living being?
Do you want cancer? At some point you need to stop doing certain things, okay?
These microRNAs, do they have any type of sensors or sensoric functions that could feel, okay, this DNA carries some cancer information, that's why I prohibit formation of it?
That is a very good question.
Yeah, I'm trying to break it down really for us to like super polite.
And I'm really sorry because of your question, it becomes more complicated. So now circular RNAs come into question.
Yeah, I already made peace with myself that we have multiple RNAs. So pull all RNAs on top of our heads.
Okay, so circular RNAs are what you expect them to be, circles of RNA. Yeah, they don't have a start, they don't have an end. They are much more stable than microRNAs, for example.
Yeah, but if you would like to sequence it in a way, do you know where to start or where to finish? Can you even sequence the circle?
You can, but you have to break it somewhere randomly and then find exon borders. But let's first talk about the function of microRNA.
Okay, let's go for it. Yeah.
And circular RNAs are one way of how microRNAs are controlled. Circular RNAs are single-stranded circles of RNA, and they also exist in each cell of your body. Okay, and circular RNAs are sometimes much bigger than microRNAs. MicroRNAs are really small nucleotide-wise; they are like 20 nucleotides, roughly.
Right.
And circular RNAs can be 1,000.
Ooh.
Sometimes this circular RNA has 20 binding sites for this microRNA. Okay, so this microRNA can bind to the bases because its complement, like here, can complement and bind several microRNAs. So let's put it all together, Iuliia. You've learned that microRNAs inhibit protein synthesis.
Exactly.
And circRNAs can sponge. We call that sponging because they can bind and they can release microRNAs. So what do you think circRNAs are ultimately doing?
Well, they're controlling the whole protein synthesis in a way.
Not entirely.
Yeah, not entirely, but are they controlling or, let's say, giving commands of some certain protein synthesis within the body? Because it looks like that. They have a pockets here of some special things, and then at some point they were like, these microRNAs should get out. But how would you say at which point should they get out? Maybe there is some, this cancer thingy that you don't want to.
The circRNA inhibit the inhibitor of protein synthesis, okay? But the most important thing for us is microRNAs, okay? That is what we are interested in because that is predicting of what proteins will come in the near future. From DNA, you cannot say this because every cell has all your DNA.
Mm-hmm.
But the mRNA is cell type specific.
Okay.
So from the DNA, you cannot say whether you have a skin cell or lung cell of your body. Well, we will talk about methylation at some point, but we will ignore this for now. But the mRNA in the cells is much more cell type specific.
That's wonderful.
Okay. So from the mRNA of your lung cell, we would be able to say, yeah, that's Iuliia's lung. That's not her muscle.
But can you say that it's Iuliia's cell and not Daniel's cell?
Yeah, because we can also talk about this. That is also one option. You can call SNPs from mRNA.
So it's not only cell type specified, that is also individual specified.
Do you know anything about Wobble, Iuliia?
Wobble?
Wobble. What?
Not at all.
Okay.
I just know how to wobble, but that's not the thing that you want to hear.
No, that's exactly the thing that is happening here. So the first two bases are really stiff and safe, but the third base can sometimes be exchanged and still result in the same amino acid.
What a beautiful life we have.
This is good because you are then resilient to mutations on that third base here. Yeah, if that mutates, it's not that bad as if one of the other two bases would mutate. Of course, your body doesn't choose which bases will mutate, but this makes a lot of mutations in your genome not harmful to your body. Okay.
Okay. Mm-hmm. Interesting.
Also, we need 3 letters here because we have only 4 bases at the mRNA, but we have, I think, 21 over 20 different amino acids. So there needs to be a translation table. Which 3 bases to which amino acids. And those are the tRNAs.
Okay. And this wobble pocket?
Yeah, this wobble is basically that the third basis sometimes does not matter. For some, if it's here an A or a G, both will end up in the same amino acid, that will end up in the same enzyme, that will work the same way.
It's something like a sticky pocket that will stick to whatever base that is near another two.
It's some flexibility on the tRNA layer, which is good because, again, even if they differ, sometimes you end up with the same well-functioning protein in the end. Doesn't harm you. But those DNA differences between both of us, the A and the T here, even though we end up with the same protein, we can sequence the mRNA and see still the difference in the letters. And we can sequence the RNA and see, oh, that's an A, that must be Daniel. That's a T, that must be Iuliia.
This is amazing for me. Yeah, but now it becomes clearer. Very nice.
We talked about the DNA at some point having a start and an end. Okay? Each chromosome has a start and an end. And that is where some enzymes start. For sequencing, we have one problem, is that we don't need one piece of DNA that we can sequence, but we need multiple. We need in the amount of millions. So how do we come from, in the worst case, from one DNA strand to millions? What do you think?
Yeah, that's exactly this polymerase chain reaction. Just because I have heard about it.
Yeah.
First of all, it's made in laboratory, so it's well-established technique right now.
Yep.
And it allows you, or maybe not you, you're a bioinformatician, right? You are not really in the wet lab, but the one scientist that are in the lab, to make copies of this specific piece of DNA. What I also know that there is some heating process involved in order to separate the strands, like to make them
denature.
Denaturation, no?
Yeah.
Yeah, exactly. So then DNA should go through this heating process, denaturation, and then Polymerase, isn't it also enzyme just because it sounds this way?
Yeah.
That is the key that helps you to do that, right?
Okay, we will start with an easy overview of PCR. So we have several steps that we do roughly 34 cycles. We want to have exponential growth in the amount of DNA. And we start with, in the worst case, again, one strand of DNA. We heat it up. So there's water, there's DNA that we put into the water.
Yeah.
And that sample we heat up to 95 degrees Celsius.
Yeah.
What this does is it pulls those two strands of DNA apart.
Mm-hmm.
So you have the same information. The bases weren't affected. The sequence is not affected by this. But you have somehow now, instead of one closed DNA, two open ones. Okay. What we also need is for an enzyme a starting point. And for this enzyme that we are working with, the open ends of the DNA that we talked about here are not enough. We need primers. Primers are small parts of DNA that we randomly make in the lab. So those are really small pieces of DNA with random nucleotide that just by chance at some point are compatible with the beginning of the single-stranded DNA. So you have single-stranded DNA where a part of it is double-stranded because a primer was fitting here.
Wait, how do you know that this random, very short piece, a primer, will end up exactly in the beginning? It could also end up being in the middle, isn't it? Or it doesn't matter? Or did I misunderstand?
No, that is a really good question. Sometimes you know the beginning. For example, if we sequence mRNA, we have a poly-A tail.
That's the thing. So that's something like a label for you to recognize that that's exactly the beginning.
And if we do poly-T primers, what happens is that it always starts at one end. That's perfect. That's easy, right?
Smart.
Yeah. With DNA, we can't do that. We need random primers. And for DNA, it sometimes will also bind in the middle. Yes. And sometimes thus we will have shorter fragments. But it can also happen that one binds in the middle and another one binds on the end. Okay.
Isn't it a disaster for you then to recognize what's going on?
No, that's easy because we can easily sort out the smaller fragments later.
Okay.
We have double-stranded DNA that we split into two single-strandeds. I will just continue with one for now because it makes it easier. And let's say that in this perfect case, we have a DNA primer that just by chance is compatible here and binds to the beginning. And now that is a starting point. Yeah. So here we start denaturing at 95 degrees, but this primer annealing happens at 45 degrees. So the DNA is cooled down. When you cool down the water, the DNA wants to bind again. Is that strange to explain it like this? But the primer then is bound because temperature sinks and the DNA is okay. So it wants to close again and we have those hydrogen bonds that will form again.
And then they cannot bind together because you already have some primers binded there.
Yeah, and that is why the other strand will not easily come here again because there are already primers that are bound. Those primers, those small fragments of DNA. After the annealing comes the extension where now enzymes come in.
Oh, hello.
Hello, enzymes. We call this one tech polymerase. It is a DNA-dependent DNA polymerase.
Nice. Sounds like a riddle. Let's see what Daniel will make out of it.
The DNA-dependent DNA polymerase can only bind to DNA and can only synthesize DNA.
You started with RNA, how it is now DNA. I mean, after we decrease the temperature and...
No. Here we started with DNA, let's say. We can also later do it in RNA, but this all here is DNA for now. Okay.
So at 95 degrees, we denaturated
the DNA. We pulled the strings apart.
We pulled the strings, then
we let it cool down.
And they bound it again. And now we're talking about...
No, they are not bounded again. They are the single-stranded DNA that are floating around now in the hot water.
Okay. Now you're talking about single-stranded
DNA. The primers.
DNA.
The primers were bound. Yeah? We cool the water down slowly and the primers bind first.
Yeah.
And the primers bind first to the single-stranded DNA.
Yeah.
And thus the single-stranded DNA doesn't go to double-stranded DNA again because the primers are on the way. Yeah. They block this.
And this tag, DNA dependent, is related to the single-stranded DNA pieces.
Where a primer is already bound and we have a starting point. Okay. This is the starting point for the DNA-dependent DNA polymerase, where now the DNA starts to go over the single-stranded DNA and puts in other DNA bases. So it's called extension because we extend the primer, basically. We put other nucleotides on top, and the tech polymerase also needs nucleotides, of course. So in this case, we have T, C, A, and G. And these are in the millions floating around in that solution already from the beginning.
Like a free nucleotides in the solution.
Yeah. Exactly. And we have the enzyme, the tech polymerase, also in that solution. We have the primers, and we put it somewhere where we can control the temperature.
What do you mean by somewhere? Thermostat?
No, it's called the thermocycler. Anyway, we have this polymerase now.
Yeah.
It docks on this primer side, takes those T, C, A's, and G's out of the solution and puts them on the single-stranded DNA and thus synthesizes the other strand of the DNA, which of course is OH at the one end, and then comes a phosphorus.
Is it in a way a self-assembly then, after you help with the temperature and with introducing the Taq polymerase?
I think the Taq polymerase does need energy. It needs ATP, but I'm not sure. That catalyzes the reaction of DNA elongation after primer annealing, after DNA denaturation. Okay?
So then these free nucleotides are flowing into the single-stranded DNA and thus making it longer.
They are compatible with the base on the other end. Yeah. So when the enzyme is done, we have then again double-stranded DNA. That is when the Taq polymerase leaves this. Then it's done. Yeah.
Okay.
We had the primer at the beginning, and then the enzyme went through and added one nucleotide after the other, made the phosphorus OH bound. Yeah.
Mm-hmm.
And made it into one coherent strand again. So now we have double-stranded DNA.
By this protocol, we doubled the amount of DNA that was in before, right?
Exactly, yes. And now comes the nice part.
Before we come to the nice part, one thing is missing for me, all these free nucleotides swimming in the solution. Can you just buy them like a powder?
Yep.
Ah, okay. Then it's clear. You have this solution and then you introduce the DNA, and then once you have a single-stranded DNA, these free nucleotides. I still would like to use the word self-assembly.
Not self-assembled. That is why the enzyme here is needed. Self-assembly would be if the nucleotides just swim here, dock on, and make this reaction here, but they are not. Yeah, this Taq polymerase enzyme is absolutely needed. If you don't add this polymerase, then nothing happens here, except that you pull it apart, primers dock, and then no more DNA gets synthesized. Because those bases here, they would dock on the single-stranded DNA, but they would not connect to each other and thus not form a coherent strand of DNA, okay?
I take my words back. Yeah, that's not a self-assembly. This is a polymerase forced assembly and doubling of the amount of a DNA and somehow transformation from a single-stranded to a double-stranded again, which is complementary then.
Exactly, it is complementary, and this happens, of course, to the other strand from the beginning, which was complementary in the beginning. Yeah? So you have the complementary of the complementary and the complementary.
And then using this thermocycle thingy, you could multiply it by whatever.
So here we go in with 2 strands of DNA.
Yeah.
And this happens to 2 strands of DNA, so we end up with 4 strands of DNA.
Exactly.
Roughly. Okay, so from 1 molecule, we go to 2.
Yeah.
And then the cool part is that this Taq polymerase survives 95 degrees. Yeah? This enzyme is stable still at 95 degrees Celsius, which means that we could just add nothing and start all over with the same sample.
And that's why the cycling.
And that's the thermocycler. Yeah? We do this roughly 30 times, and because we double each cycle, and at some point we have enough DNA for sequencing.
So it's like an exponential growth.
Exactly.
That's why it calls this chain reaction. The chain corresponds to a cycle, or is it polymerase chain? Does the chain in this PCR refers to a single-stranded DNA or to the cycle? That's what I'm asking myself. This is a double-sided, right?
No, I think the chain is here that you chain the sample several times to the
then it's kind of cycle. Cycle chain. Sounds wonderful. Yeah, you have like millions, billions of copies of the original piece that you once pulled inside of your reaction flask.
Yeah, now comes the even more interesting part. Iuliia, what happens if you design nucleotides that are not just like those simple nucleotides that we added here, but you design chemically nucleotides that don't have the OH group here? They don't. What happens? What do you think?
Without OH group. Can they have a free ending? Will they have a free ending?
The solution is?
I would not believe that they would have a free ending. Then something else will connect there.
No, it just stops. It abruptly
What stops?
And absolutely stops. The polymerase, it can't synthesize anymore. It needs the OH group for connecting the phosphorus, for connecting the next base to the other.
Can we even have the strands without like a logical ending OH in it? Is it possible that you have such broken pieces? Are they broken? Are they mutated? Like what are these pieces?
Once you have a base with a missing OH group, and the Taq polymerase still can attach this one, but it will not be able to attach the next one. Yeah, because the next one needs this OH group and the phosphorus from the next base to be attached to each other. So the DNA strand synthesis just stops abruptly with the base that has not the OH group on it.
And how can it happen that it doesn't have OH group? Or is it a theory now from a bioinformatician?
It's not a theory, it's how we sequence.
So you break it by yourself?
We make it stop abruptly at some point, yes.
Is that a correct assumption to say that in the nature, in living organisms, when we have DNAs and RNAs, they all by default have this OH? And if you want to sequence, then you force this OH somehow to go away?
Yeah, we chemically need to engineer that. That's actually quite expensive to buy those nucleotides without the OH. In nature, you don't find those nucleotides without the OH.
Okay, good. So this is forced by you experts so that you could sequence.
Yeah.
Okay, that's a bit a calming story for me, because if we would have a DNAs within us that could abruptly just stop, I mean...
Then the mRNA would be broken, then the protein resulting in that would be broken, then the protein function would be broken, and thus you would be sick or dead. Yeah.
Yeah, absolutely. What about some cancerous or some damaging DNA structures that are appearing in the body? Might it be that they don't have this OH?
To be honest, I don't know of any particular sickness or disease that comes from DNA bases suddenly lacking the OH group. It's something definitely lab grown, lab made.
All right.
Okay, so let's start. We are still on DNA, and we start with a sample that we did the PCR with. Yeah, we have this DNA strand here. With what bases are there, Iuliia?
Yeah, there is G, A, C, and T.
Good. Okay. And now, with the knowledge that we can chemically engineer nucleotides that lack the OH group, and then suddenly the DNA synthesis stops, we need another piece of knowledge. Let's say you have a gel. Is the most solid gel that you eat? I would say it's Götterspeise, for example. There's a lot of agar in it. It looks really wobbly on the table.
Yeah.
You know what I mean?
Yeah.
Yeah.
And?
Do you know something like this? Yeah, you ate it before.
Gelatin.
Gelatin. Yeah. Perfect. Good. Okay.
Did you play a word game with me right now?
No. We are doing science here. Let's assume that we have small piece of gelatin or something very similar. It's a matrix, okay? It's a wobbly matrix of something. There are holes between the molecules, but they are a defined size, those holes.
Like a membrane filter.
Something like this, like a filter, yes. But it's more like a gel.
Okay, yeah.
Okay? Imagine it being solid piece of, like a cigarette filter, maybe, but in watery solution. And we also need one more piece of knowledge, that DNA generally is negatively charged. Okay? So in solution, if you have a positively charged piece, DNA will swim towards it.
So
just
if you induce potential within your solution.
Yeah. Let's say we have a pocket, two different pockets. And one pocket, we put in long pieces of double-stranded DNA, really long, thousands of bases.
Okay.
And in the other pocket, we do very small pieces of DNA, very tiny, okay? We put them in those pockets that are inside that gel, which has a matrix with holes in it, like a cigarette filter, for example, okay? And then we put a charge at the other hand. What do you think will happen?
Yeah, then they will start to get attracted there and pull through the body of the gel towards the positive charge.
Perfect. And which one will be faster?
The short one.
Exactly. And why? Something with friction, maybe, something with the size, with the bulkiness of the molecule.
Yeah, the resistance is smaller in a way, right? How they're going through the gel.
Yeah, exactly.
Is there something else? Maybe not only resistance, no?
That's completely fine by me. Okay, this is the only thing that we needed. The one thing we needed is that we can have nucleotides with a missing OH. We can make them in the lab.
Yeah.
And if we introduce them into PCR, then suddenly at that point where the base is incorporated, after this base, the synthesis stops.
Yeah.
The piece of DNA is just stopping there. The synthesis ends, and it stays like this. Yeah, because this DNA base is still built in properly. It's stable there, but the next one will never be attached.
Okay.
Next piece of knowledge is that if we have two different lengths of DNA and we put them into this matrix gel, put a charge on it and let it run towards the positive charge, then the smaller will be faster, will be further here after a certain time than the longer piece of DNA. Okay, what do you think? If we combine those two things, how do we sequence?
If we combine these broken pieces of DNA with these ones? Yeah, I mean, how do you sequence?
We will build up slowly.
Because this piece of information that you gave now with the gel, that just tells me that we could define the length of our DNA pieces that we have in our solution.
Yeah.
But that is still not the key to start sequencing out of nothing.
It is.
It is?
Because we can define the length of the DNA piece based on the base, correct? If we have a nucleotide, a G, that doesn't have an OH end, then this piece of DNA will end at a G, okay? And this end with a G will be shorter than the full sequence. Okay, let's start slowly.
Yeah, that's a bit complicated to grasp.
Yeah, I see.
I think I'm getting there.
So
I hope you too, guys.
From the PCR, we have a lot of DNA. We have now 1 million pieces of DNA, okay?
Yeah.
And we now do a very similar thing as before, but we do it in 4 buckets. One we call the T bucket, one we call the C bucket, one we call the A bucket, and the other one we call the G bucket. In all of those buckets comes the sample, a piece of it, a part of it, a little bit, okay? So boop, 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 and boop. And in all of those buckets comes the tag polymerase, the enzyme that we talked about, the elongation of the chain. And in all of those 4 buckets, T, C, A, and G, comes the nucleotides. But also, and here's the trick, in each of those buckets comes 1% of not only the typical T, C, A, G nucleotides, but in the T bucket, we put roughly 1% of T nucleotides that doesn't have the OH group.
Okay.
Okay? So roughly 1% of the nucleotides in the T make it suddenly stop at that point. Okay? We do the same with the C, where roughly 1% of the C nucleotides make the polymerase chain reaction to stop. We do the same with the A. Roughly 1% of A's in that solution will make the DNA elongation stop at a nucleotide. And roughly 1% of G's will also stop the PCR.
Okay.
You keep track of those, which one is what letter, and you do a typical PCR like we did before, with the denaturing of the DNA, with the primers, with the annealing then of the primers, and then you have the tag polymerase that starts to synthesize the DNA. But at some point in the T, we have a T built in just by chance because it's 1%, which stops it, which stops the elongation of the DNA, okay? This DNA piece here, let's say it is 1,000 nucleotides long, okay?
Yeah.
And a time point, so where in the sequence the T is that stops the synthesis, is completely random. We cannot control it. We added this at the beginning, so at some point it will stop here. At some point it will stop at the very end, maybe because it's a T. At some point it will stay here, blah, 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 blah. And we have, after a PCR, we typically have millions of DNA pieces, because we made it several cycles, so it was exponentially growing. But we, in the end, we have a random distribution of length of these. At some point stopped by a T. It always stopped at a T. The original sample, that has the full length, at this point is of course only a small fraction of the total DNA in that bucket. Because once we did the PCR, most of the DNA in this bucket is the newly synthesized, with at some point being stopped by a T. It is only a small fraction of the original DNA that is still in the bucket afterwards.
Okay.
And now we still keep track of the T, C, A, and G bucket, but we do the same with it. We all do a PCR where at some point randomly it stops. And now we go again into this matrix gel. Okay? We put this T in a pocket. We do the same with the C, with the A, and with the G. We again put a charge on it. So what do you think will happen?
Yeah, depending on the length, you will get different speed of. It's not really the pockets. It's like nucleotide chains.
Yeah, length of chain. That's the case, yeah.
Anyhow, you will have a random distribution in each of them because if we are talking about the pocket T, in every strand, the T will happen randomly somewhere in the beginning, at the end, and so on. And then in this pocket, you will have a number of different lengths, right? Like many of them. Some of them shorter, some of them longer.
Absolutely.
Yeah, then I'm still having complications. What is the sequencing and how does it go? Because now you basically multiply it and then you have a combination of whatever, like a huge randomness within your pockets.
Exactly.
And I mean, no clarity so far.
Okay, so we said that in this pocket there are molecules which have different sizes, and the sizes define how fast you are in the gel. Yeah? We stop when the first DNA piece here gets through the gel. At that point we stop. We have the maximum resolution. And we still keep track of which one is what base. And as I imaged here now, we have again several speeds for each bucket because we have several length of DNA parts.
Yes.
The DNA pieces that stopped at different parts of the PCR. And if you now do a result of this, at this position here, at the first, the slowest was this G. So we have here a G. The second slowest fragment was somehow part of the C. So we have a C. Right after this comes here a T, and then exactly. That is how we see. So it's just based on the different speeds of the DNA fragments resulting from the PCR that at some point with a specific base stops, and thus dictating the length of the DNA and thus dictating the speed in the gel.
You could fish out the exact position of nucleotides within the whole sequence. I didn't see it coming.
The best thing about it is that this, how we explained it now with the 4 different buckets for T, C, A, and G and the 1% of the without the OH nucleotides, that is the most used sequencing technique worldwide.
But is that this Sanger technique that
is
you
the
want
Sanger
to say?
technique. One thing that I need to mention also is that we don't just add the nucleotides with the OH group missing, but those nucleotides also have attached to it on the side, so it doesn't affect the reaction. But on the side, they have something like a glowing thing, just a marker that when you shine light on it, you see it more clearly.
Is it GFP?
No, it's not a whole protein that we attach to bases. That would make it too clunky. But it's something similar to this, where we attach just a defined signal onto that base that doesn't affect the reaction, just makes it more easier on the gel than to separate them.
Then you're using something like fluorescent lamp to check out.
Exactly. In the beginning, it was made with radioactive bases, and then you would check with a radio signal, not with the radio from the car radio, but the radioactive signal from the gel where it is the most intense.
And is that one of the first sequencing techniques that were discovered?
That is Sanger. Frederick Sanger invented it. Again, it is the most used sequencing technique today.
Still, so it's one of the firsts, and it's still on the market.
Yes.
Are there some other sequencing techniques?
Yes.
Okay.
There are multiple ones which are cheaper per gigabase, but the interesting thing is if you give me one sample with 1,000 DNA bases and you give me $10, then I will sequence it for you. 10 euros.
How well is the result at the end? So is it kind of guaranteed? Are there some big errors? Because it sounds like super cheap, and from this perspective, why would I go to any other sequencing techniques? I mean, I can just pay and go.
So with Sanger, you have maximum resolution in length here. This gel cannot be 3 meters. You have a specific resolution that you can achieve. And at our laboratories here, you can roughly sequence up to 1 kb, so 1,000 bases in sequence. But you can only sequence one fragment, one PCR result here. One thing, one read comes out of it. That is not a high throughput sequencing technique, but it is important, for example, when you want to diagnose specific cancers, and those specific cancers are dependent on certain sequences in your DNA, and you know the sequences. Then you can, with a primer, with a very specific primer in the PCR of the whole genome, you can pick those out, and then you can just sequence this short fragment and see from those 1,000 bases which 2 or 3 are different, and thus are you sick with a more aggressive cancer or less aggressive cancer.
And this is exactly how these COVID tests worked for us, right? With this PCR, isn't it?
The PCR tests. So the COVID tests used real-time PCR, where you have those nucleotides with a shiny thing on them, and very specific primers, COVID primers. Okay. And if those primers attach to your sample, which is your DNA in the mucus or blood or wherever, then the DNA gets made. There is more DNA. There's more shiny, and the brighter the signal, the higher is your signal. That also explains you sometimes get a value out of those PCR tests. And the low value is a high amount of virus, and a high value is a low amount of virus. That comes from how many cycles you needed until you could detect the signal that, yes, there is more DNA coming. So there must be a COVID sequence somewhere in your sample. And the least cycles you need, the more DNA was in there from the beginning. Okay.
Yeah, amazing. I think that for fundamentals now we really came to a good point. We named the technique that there is a Sanger sequencing, which was discovered. Do you have a year?
The Sanger sequencing technique was developed by Frederick Sanger and colleagues in 1977.
1977. Okay.
Yes.
Yeah.
How safe it is, you ask? It's around 99.99% safe.
How it is for bioinformaticians? Is it safe enough, or you would like to have something even safer? Because for me, I would say, well enough. But what about you?
99.99% is good. Let's say it's more for the clinic, where you have something where you know exactly which gene could or could not be modified to make it more interesting or to change the treatment. There you have, let's say, gene B on nucleotide 13. If that is a T and not an A, then you would need more aggressive therapy.
Okay.
That is where sequencing comes in, Sanger sequencing comes in, for example. The oxynucleotide concentration is roughly 100-fold higher than that of the corresponding D-deoxynucleotide. D-deoxynucleotide is without the OH group.
You lost me with this sentence.
So the T is also in there, but also the T with a missing OH group. And that concentration is roughly 100. So the T is 100 times there, 99 times, and the T with the missing OH is 1 time there.
Yeah, actually, that's still the point. Why 1%?
Because you need some amount of DNA to make it through the PCR to almost the end, because otherwise you wouldn't be able to detect the 2 or 3 DNA pieces here. You need hundreds of each in each point to detect them, to have a strong enough signal at that point. So you need it to be only 1% stopping, so that most of the DNA gets through later points. If you make it 3 or 5%, then you would only be able to detect half the length, because it would break up more early, because the chances are higher that it will build in an T with a break signal rather than a continuous signal, basically.
I think this is a very important point, because my first impression would be, come on, 1%, like add half of it, and then be done faster, but not at all.
Yeah.
Then you're really losing in length, and then you cannot sequence half of it, maybe.
So the PCR cycles are not that long. If you make 32 PCR cycles, that's half a day, not more, because on most of the denaturing it takes only a couple of seconds.
Yeah.
You don't have to cook a liter. It's only like a microliter.
Yeah, but this half a day, also wet lab scientists should be there and take care of it.
So you pre-program the PCR cycles, temperature, time, next temperature, next time, how many times you repeat what. You put all things, including the protein, the tech polymerase, you put it in at the beginning before it starts. And at the end, the thermocycler can also cool it to 6 degrees, so store it safely until the next morning.
Okay, so it's kind of an automatized procedure already.
Thermocyclers are widely used in laboratories, because a lot of reactions or enzymes you can control with temperature, and that makes it really handy.
Yeah, and when you have these pre-programmed protocols that you could put inside, then it's just golden.
Sanger sequencing, it's not the most used sequencing technique in our lab. We do offer Sanger sequencing still for this very specific questions. But the problem in that case is that Sanger sequencing, we are only able to sequence one piece of DNA at a time. For the next piece of DNA, you would need to repeat that procedure all over. And now we are at sequencing techniques where we can sequence millions or even billions of DNA pieces at once. Highly parallel. But that is just a spoiler for the next time.
I'm curious about new sequencing techniques that we are going to discuss. And I promise not to Google, so that you're getting the first-hand experience and also sincere questions of mine.
Yeah. Next time we will arrive in the year 2000.
All right.
Yeah, we will make a time switch forward. And we are looking forward to your feedback.
Yeah, thank you very much for listening in, and see you next time.
Bye-bye.
Turn into Explain today and discover the potential of the next generation sequencing.
Shall we eat? Is someone hungry on the other side here?